This session is being recorded now. The session is being recorded. Good morning, ladies and gentlemen. We are now going to proceed and finish off our lecture on morphology. But before we proceed, I just want to do a quick recap of what we talked about, some of the things we talked about yesterday. This would include the classification of microorganisms based on, I beg your pardon, can I take a call, please? It's important, just hang on for a minute. Thank you very much. Hello, Prof.
But
Okay. Okay. All right, thanks. No, I'm also in the middle of a lecture, so I'll just run with it after the lecture. Yes. Oh, okay. Thanks, Prof. Okay. Bye. Bye. Hello, guys. My apologies. Sorry for the long interruption. Let us proceed. My apologies. What I was talking about is just for us to do a quick recap and talk about the positions of the flagella first before we go on to our talk of the day, which is the spore. So we did mention things like monotrichus, lopotrichus, and then amphitrichus and peritrichus. I just wanted to recap with you on these types of flagella arrangements. All right. So we can we can move on to the next one, which I want us to look at this morning. Right. We were talking about the endospore and its importance. I remember I did talk about the fact that it has got, only between 10 to 30% in terms of water content compared to the vegetative cell. And that gives it an advantage whereby it will not be able to grow. Rather, it goes into a dormant state. And that dormant state is quite stable and it will make the spore survive very harsh conditions. I said this is how the cross section of the spore itself looks like, and it's a structure that would do well to understand how it is structured. You can see that from the spore cell to get to the outside, it is really protected by a series of layers. Hence, these layers here confer the resistance and the rigidity and toughness that the spore has got in order for it to be resistant and for it to survive very harsh conditions. So I was talking about another acid, which I said that acid is called dipicolinic acid, and that is where we left off, I think. Yes, and this is the structure of dipicolinic acid, the chemical structure. It's quite easy. You can see it is a benzene ring with a nitrogen at the para position, and then two um, carboxyl active groups, which are both um, at the meta position of the benzene ring. So these then join together, or they are bonded together to another uh, uh, moiety of the dipicolinic acid via calcium ions. And as the as the calcium is incorporated, it makes the dipicolinic acid chain complete, and then the strength of the endospore is enhanced. So why are we talking about dipicolinic acid? Because it is characteristic of endospores. It gives the character of the endospore. And then again, it is a major component. You can see it makes up between 5 and 15% of the dry weight, just one component, making that much contribution to the dry weight of the endospore. Uh, what is its job? It is there for heat resistance of the endospore. So quite an important acid for us to remember, dipicolinic acid. Right, and then some spores were found to exist, for a very long time, and according to reports, some spores which were over 250 million years old were actually revived and they were able to grow. This is quite amazing to see how organisms really can, survive. For me, I had an experience whereby um, there was a spore that was preserved in a lyophilized format, that is a powdered format, and then frozen in the lab where I was doing my undergraduate studies. And then I was asked to check out those spores. And at that time, the spores had stayed for quite a long time. They were preserved exactly 26 years before. So nobody gave them a chance that will they be still viable? Because normally, in the lab, if you're working with bacteria and if you're working with fungi and so on, if you try to keep them, especially using other short-term methods, they will only survive for about a week or two. After 2 weeks, it's a give and take. You may find them able to grow or they may not grow. But this spore, which I'm talking about, was over 25 years old. And then I took it and revived it. And to my surprise and to everyone else's surprise, the organism grew very, very well, as if nothing had happened to it all this other time when it was lying dormant. So the spores really can protect the organisms for quite a long time. Some of the spores were actually preserved in salt crystals during what is known as the Permian age, and some of it was actually revived from brine deposits, which are some salt deposits, and so on. So you find that it is actually possible to naturally preserve the spores, and they just wait for the right kind of conditions for them to grow. What happens for them to grow? They are actually triggered for them to be produced, for sporulation to take place. It is triggered by conditions that are harsh, in other words, suboptimal conditions. In other words, I'm talking about conditions that are not favorable for growth and for the vegetative state to proliferate. So what happens is those starvation conditions, or those conditions of high pressure, conditions of uh, cold, excessive heat, and so on, they tend to put a pressure on the organism which can form the spores. The organism will sense the danger, and then it goes into sporulation quickly. And the whole process is quite quick. It takes about 8 hours for a spore to, from the point of formation to the end, right? And then what happens now for the spore for it to become a vegetative cell again? It returns to the vegetative state after it germinates. We say it germinates. Remember the example that I talked to you about? I gave you an example of a seed. That's more or less the context in which you can think of a spore. So it germinates, and the germination doesn't take time. As long as the conditions are good, it can germinate within minutes. And what does it do when it germinates? Um, the covering coats crack, and then the spore cell inside then begins to grow into vegetative bacteria, and as a result, the organism starts to live again, right? So uh, the sporulation stages are normally investigated or studied um, when they look at those other types of mutants within a population, those mutants which do not form spores. In other words, they've changed a little bit in their genetic makeup so that now they are unable to form spores. Then the scientists would see or decide or determine where the sporulation is blocked. Then they make that a series of blockings and investigation. The blocking is there now. They investigate others. They see a block on another point. Then that, if you combine the whole series of investigations, they will give you an indication of how the spore has been formed from the time it began to the time it matures. So they noticed that actually it's about 200 genes that are involved in the sporulation process. Now, I expect you, like I said before, to know the sporulation process, to know the structure of the spore, and to know about dipicolinic acid, its importance, and so on. So we will now move on to talk about the the sporulation itself, how it takes place in the stages. There are a total of 8 stages, starting from 0 to 7, and I'll quickly just go through them. I want you to study this by yourselves. There's a stage 0, whereby the cell begins to prepare for sporulation, and then with stage 1, the um, the DNA becomes more dense. In other words, now it's preparing to separate out. And then what happens is there is an initial spore that starts to develop um, in stage 2. And then in stage 3, you get what is called engulfment, whereby the new spore that is being formed will be engulfed by the larger part of the cell that remains vegetative when the spore begins to form. And then you then move on to what we call a 4 spore. And with the 4 spore, it's almost complete. However, it is not strong at this stage. And then you get now to the formation and strengthening of the outer layers like the exosporium and the cortex. This is now going to be stage 4. And then with stage 5, you get the addition of ion in the, of calcium. That's calcium 2 plus ions. And these, remember, they are part of the chain of dipicolinic acid to form a solid wall, which gives that resistance I've talked about. And then the spore now has to go through stage 6 and 7, where it matures in stage 6, and then it is released in stage 7. By release, I mean that it is freed from the mother cell and it exists as a free living spore, right? I also would want you to watch the video also. It is in the it is on Blackboard in the video section. You can have a look at the animation there to see how the spore is formed. So that's what I wanted to share with you up to yesterday, but we couldn't get through to sporulation, which is what we have done this morning. So we can take a minute's break or so as we move on to the next session of the day. Before I do that, I will just ask Ms. Chipeta if there are any questions in the chat or if anyone is raising a hand. Yes, you can go ahead, Ms. Chipeta.
Good morning, Doc. We have a question concerning the WhatsApp group for this subject. Do we have any group?
Okay, thank you very much for the question, a very pertinent one. Basically, what was decided, okay, let me just say it as it is. Because what was decided was that if you notice the previous years, we used to have a WhatsApp group for each and every subject. Then it so happened that the communication that happened on the WhatsApp groups, eventually people noticed that it was some of it was actually open to abuse. Students were abusing other students, and students were, so many things happened. Some were being rude even to the members of staff. And so eventually it was agreed that, we minimize the number of groups that we are going to have. And it's a departmental decision because the WhatsApp platform is not really an official platform. It is an additional aspect which we just make use of because it is quick for us to reach our students. So the compromise was made to say we will still have WhatsApp because it's important, but we will monitor the kind of talk and whatever that's going on there. And then also we will minimize the number of groups. In other words, if there's just one group for food tech and for food micro, now that group, if one wants to post something regarding micro, food micro one, you would just say hashtag MIB150X or hashtag MIB150S or hashtag FOT150S, something like that. Then you say what you want to say after that. So we don't have a separate group for micro on its own. This was decided after a number of facts, a number of events, and the groups then were reduced so that, also there was an outcry from some of the students to say that they've got too many groups, and others were saying they're comfortable with it. But eventually, a compromise was made to say, let's then have fewer WhatsApp groups. So this year, the WhatsApp groups are actually fewer than the past 2 years.
Okay, Doc, we have a second question from Dylan.
Yes.
He's asking, how does food microbiologist make sure that spores do not form when trying to heat sterilize food or containers? And then he's basing this on the basis that bacteria form spore under extreme conditions.
A very good question. Thank you very much, Dylan. What happens is that when you are in the industry and you are producing food, we make use of knowledge that we have. You are actually going to acquire that knowledge by the time you finish with this course. We also have to go through some processing technologies, and then in those technologies, we mix the technology with the science. So what happens is that we know for sure that if you are going to, for instance, apply heat and a certain amount of pressure to the organisms, they will be killed by that. So in producing food, we do what we call a D12 heating process, or thermal processing of the food that you are producing. Now, this is a situation whereby we just generally call it commercial sterilization, or we call it a Clostridium botulinum cook. An organism like Clostridium botulinum is actually very, very resistant. Its spores are quite resistant to extreme pressure and to high heat. So if you are able to eliminate Clostridium botulinum, the other spores that might be there or that might be activated, they will die off. Because if you apply heat to spores, you actually activate them, that they start to germinate. But the heat that you apply when you are processing is so intense, it goes way beyond what the spore can tolerate. Excuse me. So you asked that the spores are formed during extreme conditions, right? Spores are formed during extreme conditions, that's right. But when you are processing the food, you are processing it under even worse conditions, to such an extent that no organism will basically survive that. So if you go back to what I was referring to when I talked about the D12 processing, that means you are going to reduce, theoretically and also practically, the number of surviving organisms. 90% of them will be reduced 12 times. So you do 12 cycles where you are reducing the population that is existing by 90%. So if you look at that, if you consider that the normal appearance of food, when it is appearing to be spoiled in our eyes, at that juncture, the count, if you do a count on the population, it is round about 10 to the 7, 10 to the power 7 colony forming units, or viable bacteria. So at that concentration, which is 10 to the power 7, if you then have to reduce the population 90% at a time, but repeating it 12 times, you will see that theoretically you will not be able to have any organism surviving, uh, maybe after the 8th repeat or the 9th repeat. But in industry, you do it like 12 cycles. So even the most resistant spores like Bacillus stereothermophilus and Clostridium botulinum, they will not survive this process. Hence, the food you produce will be safe at the end of the day. So yes, indeed, pressure or extreme conditions will activate the organisms that can form spores to go into sporulation. But at the same time, when you are doing production, you don't give it the conditions that will allow it to sporulate only, but you go way beyond, so that the spores themselves, even if they try to be formed, they will be decimated again with that uh, production process. Do we have another question?
Yes, we have a question from Lindelwa and Ayanda. It's the same question. So they are both doing second year, but also in the food micro one. They are
Yeah.
confused about the practical date of which they've missed the first section, and they have also sent an email to Dr. Henning, but she has not responded.
Okay. The practicals, let's deal with them offline.
Okay.
I think Dr. Henning would respond to that. I will talk to her, but can I just ask the students to forward to me the email that they sent to Dr. Henning? I am sure, or I hope that we will be able to, um, try to come through for the students. They'll just send an email to me to say what happened or why they were unable to attend the practical, and then I will um, uh, contact my colleague, Dr. Henning. I'm sure she will respond to that. Um, so we can take it offline so that we solve it offline. Thank you.
Thank you, Doc.
So there are no further questions.
Yes, Doc, no further questions.
All right, thank you very much. Um, what we can do now is I am not going to take much time. I'm just going to introduce our next lecture and then we call it a day because there are certain things that have come up which are quite urgent according to the instruction that I got from the HOD just as the lecture started, the phone call that I received, but also even from yesterday, I had promised you guys that we're not going to go deep into the lectures today. We just wanted to finish off sporulation. So for now, we can um, talk about um, microbial growth. I just want to quickly um, introduce prokaryotic growth, and then we call it a day. I'm loading the file. It's almost 50% done now. Right, so when we talk about bacterial growth, we can refer to that as prokaryotic growth. So in terms of prokaryotic growth, we will be looking at what definition that we have in terms of growth, because we should be able to see the difference in growth of, let's say, an animal like cattle and the difference between its growth and the growth of bacteria. So when we talk about the growth of bacteria, we are talking about how the bacteria multiply or how they increase in number, right? So I'll just introduce here to say that bacterial growth takes place in a process that most of the times it takes place in ways that are known at the moment. And if you look at things like binary fission, and if you look at binary fission, budding, and fragmentation, right? These are ways in which the bacteria can grow. But the most important way in which bacteria grow or reproduce is called binary fission. So if I ask you, the way by which bacteria grow. At this stage, I am expecting you to talk to me about binary fission. So what is this binary fission? It is actually an asexual reproduction in which the cell or the organism separates into two cells. So the cells enlarge and divide to yield two daughter cells of equal size. This is the most common form of reproduction when it comes to microorganisms. By far, binary fission is the most common. But you get other types of organisms that bud and others that fragment. So, for instance, when you talk about yeast, they do what is called budding. So by budding, it's like an outgrowth or a shoot comes out, and that outgrowth, it's actually something that continues to enlarge, and it enlarges until it forms a completely new organism, and then which clips off and separates out. So in this instance now, the daughter cell is quite smaller compared to the mother cell. But in terms of binary fission, the cells that come up, the daughter cells, they are of equal size, comparing the daughter and the mother cell after binary fission. So binary fission itself has this effect of doubling all the time. So we are going to do a few calculations based on how bacteria go through binary fission, because they mathematically double after a given period of time, which we call the generation time. If you start off with one viable cell, when its generation time is complete, you'll see that it divides and forms two cells. When another generation time is completed, the two cells become four. Similarly, the four cells having gone through another cycle, they then change and become eight. 8 becomes 16, and so on and so forth. So that's binary fission. We are going to go more into this one, but suffice to say that a good number of yeasts go through what is known as budding, and then other organisms like the filamentous bacteria, they do what is called fragmentation, whereby they just appear like they are breaking off. But when they break off, each bit that breaks off or that fragments will later just close off and become a new organism, right? So that's fragmentation. And now I'm going to bring in the last slide of this morning, right? Growth of bacteria will take it to be an increase in the number of cells. It is not necessarily an increase in size, right? And the generation time, which I was talking about, is the time that is required for the number of cells to increase. So a given population, for it to double, for instance, that time that elapses before you get a doubling of the population, we call it generation time. And what is the generation time for organisms, for prokaryotes? It is a minimum of 10 minutes, and others that are very slow can actually take days to double in their number. So the fast growers take minutes, and the slow growers take days before the number doubles. Then we will also be looking at things like what is the growth rate, right? So the growth rate would actually be an indication of the cell number over time. And I indicated there that the D cell number over time, or the D cell mass over time, which is the number of cells or the daughter cells given that you get after a given time. So if you're talking about rate, you have to include a factor of time, how long it is taking for the biomass to increase. Right. Having introduced prokaryotic growth and saying that we are going to concentrate on binary fission in what we are going to talk about, I will then take off next Friday on, we'll take up from the slide of, from here, we just go on to talk about the binary fission. So guys, study ahead of me and understand what binary fission is all about, and also the bacterial life cycle, whereby we're going to start from the lag phase, the log phase, the stationary phase, and the death or decline phase. Uh, Study those things so that when we meet again, you'll be able to understand better as we go through that lecture. Thank you very much. Do we have further questions, Ms. Chipeta, before we close?
Yes, we have a question from Faith. Do we have class on Friday? That's Friday.
Yes, we have a class this Friday. I'll set up the new link because I noticed that the link that I had put there, the recurring link, I think it expired. So I'm going to set up another link and share with you guys. We'll meet again on Friday. That is correct.
Okay, Doc. A question from Sianda. Is the term sporulation the same as sporogenesis?
Thank you very much. Those terms are synonymous. Thank you. They are the same. Sporogenesis and sporulation, they are the same. The genesis, you will see that it's taking us to the concept of how the spore is formed, and that's exactly sporulation. The terms are similar. Thank you very much.
No further questions, Doc.
Thank you very much, Ms. Chipeta. Thank you very much, guys, for attending. Let's meet again next Friday and do this thing over again. And just study ahead of me so that when we move, we also move with more speed and understanding. Um, enjoy your day, and God bless. I'm now stopping the recording.